pyruvate dehydrogenase complex needed for the conversion of pyruvic acid to acetyl-CoA is located in matrix of mitochondria. S phase, as we know it is called as synthetic phase, is the part of the cell cycle in which DNA is replicated. We know that cosmerium is the one which is non-motile and it is a fresh member of chlorophyta. So I am back with part 2 of this Karnataka CET 2010 biology paper analysis. Moving on to question number 21. Identify the pair that exhibit cercinate vernation. So you come across leaf like this. So this is called as cercinate vernation. A. Equisetum and Selaginella. B. Silotum and Rixia. Then C. Rixia and Nephrolepis. And D. Nephrolepis and Cycas. So the pair that exhibits cercinate vernation among the four is Nephrolepis and Cycas. They exhibit cercinate vernation like this. So the leaf will be like this. So the cercinate vernation is the manner in which the frond, here we don't use the word called leaf, we use the word called frond, emerges. As the fern frond is formed, it is tightly curled. So you can see the curling, so that the tender growing tip of the frond remains protected. So it is Nephrolepis and Cycas. Option A, Equisetum and Selaginella is wrong. Option B, Silotum and Rixia is wrong. And option C, Rixia and Nephrolepis is wrong. And option D, Nephrolepis and Cycas is correct. So option D is correct. So pyruvate, question number 22. Pyruvate dehydrogenase complex needed for the conversion of pyruvic acid to acetyl-CoA is located in option A, intermembranous space of mitochondria, B, matrix of mitochondria, then C, cytoplasm, and D, grana of chloroplast. So the pyruvate dehydrogenase complex needed for the conversion of pyruvic acid to acetyl-CoA is located in matrix of mitochondria. The liquid-like substance, what we use the word called matrix, is found in the matrix of mitochondria. So option A, intermembranous space of mitochondria is wrong. Option B, matrix of mitochondria is correct. Option C, cytoplasm is wrong. And option D, grana of chloroplast is wrong. So option B is correct. Moving on to question number 23. The presence of corollary corona, sagittate anthers, and dumbbell-shaped stigma are the characteristic features of Hibiscus rosa sinensis, which belongs to Malvaceae. Then Musa paradisiaca. Then C. Ravenella madagascariensis. Then D. Catharanthus roseus. Here we find that the flower is bisexual, actinomorphic, gamosepalous, that is the sepals are fused, gamopetalous, that is the petals are fused, and the anthers are sagittate anthers with adherent to stigma carpels, that is gynoecium, two carpels which are united by a single style. All these are the features of that is observed in Vinca rosea, that is Catharanthus roseus, which belongs to a family called Apocynaceae. So option A, Hibiscus rosa sinensis is wrong. Option B, Musa paradisiaca is wrong. Option D, Ravenella madagascariensis is wrong. Option D, Catharanthus roseus. So Vinca rosea is also called as what? Catharanthus roseus. So option D is correct. Moving on to question number 24. Every time when the dosage of a drug has to be increased to achieve the same kick that initially occurred in response to a smaller dose, this condition is known as option A, tolerance, option B, rebound effect, option C, addiction, and option D, withdrawal symptoms. Every time when the dosage of a drug has to be increased to achieve the same kick that initially occurred in response to a smaller dose, this condition is known as tolerance. It is known as tolerance. What is tolerance then? It is a condition in which the body gets used to drugs. Like continuously if we keep on taking medicines, then later when we take the medicines, that will not have any effect on the body. So that condition is called as tolerance. So tolerance is a condition in which the body gets used to drugs due to drug addiction, and the person's reaction to drugs get reduced. Like if you continuously take painkillers, then when you have pain, if you take painkillers, it will not have any effect on the body. So body gets used to the dosage and produce no reaction. So option A, tolerance, is correct. Option B, rebound effect, is wrong. Option C, addiction, is wrong. Option D, withdrawal, is wrong. So option A is correct. Moving on to question number 25. DNA gyrase, the enzyme that participates in the process of DNA replication, is a type of option A, reverse transcriptase. Option B, DNA topoisomerase. Option C, DNA polymerase. And option D, DNA ligase. DNA gyrase, as we know, it is an ATP dependent type 2 topoisomerase, which is an enzyme which plays an important role in the negative supercoiling of double-stranded closed circular DNA that is found in bacteria. So the DNA gyrase, the enzyme that participates in the process of DNA replication, is a type of DNA topoisomerase. So option A, reverse transcriptase, is wrong. B, DNA topoisomerase, is correct. Option D, DNA polymerase, is wrong. And option D, DNA ligase, is wrong. So option B is correct. Moving on to question number 26. Mendel found that the reciprocal crosses yielded identical results. From that, he concluded that option A, sex plays a role in deciding the dominance of a trait. There is no option B, there is independent assortment of traits. Option C, sex has no influence on the dominance of traits. And option D, there is no dominance of any trait. What is reciprocal cross? A cross with the phenotype of each sex reversed as compared with the original cross to test the role of parental sex on inheritance pattern is called reciprocal cross. So Mendel found that the reciprocal cross yielded identical results. From that, he concluded that sex has no influence on the dominance of the trait. So option A, sex plays a role in deciding the dominance, wrong. There is independent assortment, wrong. Sex has no influence on the dominance of traits, is correct. There is no dominance of trait, is wrong. So option C is correct. Moving on to question number 27. Compare the statements A and B. Statement A, synthesis of DNA takes place in the S phase of interphase. Statement B, every chromosome during metaphase has 2 chromatids. Choose the correct description. Synthesis of DNA takes place in the S phase of interphase. Correct. S phase, as we know, it is called a synthetic phase, is the part of the cell cycle in which DNA is replicated and which occurs between G1 and G2 phase. So statement A is correct. B, every chromosome during metaphase has 2 chromatids. Correct. In metaphase, during the metaphase stage of mitotic cell division, each pair of bivalent, that is 2 chromosomes, 4 chromatids, align, they arrange themselves on the equatorial plane. It is called as metaphase plate. So it is also correct. So both the statements are correct. Option A, statement is correct and B is wrong, is wrong. Statement A is wrong and B is wrong, correct, is wrong. Both the statements A and B are correct and A is not the reason for is wrong. Both the statements A and B are correct and A is the reason for B. So option D is correct. Moving on to question number 28. Match the animals listed in column 1 with their nature of blood listed in column 2. Choose the answer which gives the correct combination of alphabets of the 2 columns. A, man. B, earthworm. C, cockroach. D, frog. P, plasma and cells are colorless. Plasma is colorless and nucleated RBC. R, plasma is colorless and enucleated RBC. S, plasma is red and nucleated colorless RBC. T, plasma and RBC have hemoglobin. So man is plasma is colorless and enucleated. Then earthworm Earthworm plasma is red and nucleated colorless RBC. That is yes. Then cockroach, P. Plasma and cells are colorless, right? Then frog, plasma is colorless and nucleated. That is Q. So AR, BS, CP, DQ. Go for the first option, AR. Here AS, wrong. Here it is AR, correct. Here it is AT, wrong. Here also AP, wrong. So obviously this option is the correct one. BS, here you can see BS, correct. CP, correct. DQ, correct. So option B is the right answer. Moving on to question number 29. During lactic acid fermentation. Fermentation is of two types. One is alcoholic fermentation, the other one is called lactic acid formation. In alcoholic fermentation, we can see the release of carbon dioxide along with ethyl alcohol. So, in lactic acid fermentation, as you know, it is an anaerobic metabolic process and carried out by few bacteria like lactobacillus and even animal cells. So, enzyme called lactate dehydrogenase participates in this process and converts the pyruvate produced during glycolysis into lactic acid. So, here neither O2 is used nor CO2 is produced. So, option A, neither O2 is used nor CO2 is liberated is correct. O2 is used, CO2 is liberated is wrong. And option C, O2 is not used, CO2 is liberated is wrong. And option D, O2 is used, CO2 is released is wrong. So, option A is correct. Moving on to question number 30. Which one of the following is not the function of insulin? As we know that pancreas is an exocrine gland which acts both as an endocrine organ as well as exocrine organ. The endocrine part of pancreas called the islets of Langerhans produces two types of cells, alpha cells and beta cells. Alpha cells produces a hormone called glucagon and beta cells produces an hormone called insulin. And you know, both have a different function to perform. Insulin will bring down the glucose level to normalcy when there is a rise in the glucose level. So when there is a fall of glucose level in the blood, then glucagon comes to the rescue and converts back glycogen into glucose. Insulin converts glucose back into glycogen, thereby balancing the glucose level in the blood. So insulin, as I said, it is produced by the beta cells of islets of Langerhans. This insulin acts on hepatocytes, that is the liver cells, and also adipocytes, and increases the hepatocytes' capacity to absorb more of glucose, cellular glucose and its utilization. So, converts glucose into glycogen. Glucagon converts glycogen into glucose. So, which one among the following is not the function of insulin? Increase the oxidation of glucose in the cells is the function. Increases the permeability of cell membrane to glucose is the function. Initiates the formation of hepatic glycogen from excess of glucose, correct. Initiates the conversion of glycogen to glucose is wrong. So, option D is not the function of insulin. Moving on to question number 31. Which one of the following is a unicellular non-motile desmid? Option A, Clostridium. Option B, Chlorobium. Option C, Cosmerium. And D, Chromatium. We know that Cosmerium is the one which is non-motile and it is a fresh member of Chlorophyta. And this Cosmerium is relatively large and it is unicellular, having no locomotory organelles, and hence they are non-motile. So the following, which one of the following is a unicellular non-motile desmid? It is Cosmerium. So option A, Clostridium, wrong. Bacteria, it is Chlorobium, wrong. Bacteria. Cosmerium is correct. Option D is wrong. So option C is correct. Moving on to question number 32. Some of the steps involved in the production of humulin are given below. Choose the correct sequence. First one, synthesis of gene DNA for human insulin artificially, culturing recombinant E. coli in bioreactors, purification of humulin, insertion of human insulin gene into plasmid, introduction of recombinant plasmid into E. coli, extraction of gene product from E. coli, that is extraction of recombinant gene product from E. coli. So some of the steps involved. Choose the correct sequence. So first one is what? Synthesis of gene from human insulin artificially. Then second is insertion of human insulin gene into plasmid. Then comes introduction of recombinant plasmid into E. coli. Then culturing recombinant E. coli in bioreactors, then extraction of recombinant gene product from E. coli. So, first one is I, then 4, then 5, then 2, then 3, then 6. So, this is the option. So, first one I, here it is also I, here 1, here also it is 2, so this is wrong. Here it is 3, it is wrong. So, here it is 1, correct. Go for the next one, 4, here it is 1, here it is 5, so this is wrong. Here you can see 4, then 5, correct, then 2, correct. This is 5 and this is 6. This is 6. So, 5 and this is 3. So, option D is correct. So, option D is correct. Moving on to question number 33. Cockroaches can climb smooth or steep surfaces due to the presence of adhesive pads found on the tarsus of their legs. They are called option A, tibia, option B, plantulae, option C, aroleum, and option D, pretarsus. Actually, it is aroleum. It is the adhesive pad of the cockroach foot engaged when the legs are pulling and used in scaling and adhering to the smooth surface. So, it will help. It is acting as what? Adhesive pad. Adhesive means to attach itself to the wall, smooth surfaces. So, it is aroleum. So, option A, tibia is wrong. Option B, plantulae is wrong. Option C, aroleum is correct. And option D, pretarsus is wrong. So, option aroleum is the correct answer. Moving on to question number 34. Gastrula has a pore which is known as option A, gonophore, option B, blastopore, option C, oospore, and option D, zoospore. We know that gastrula has a pore which is known as blastopore, an opening called blastopore by which it communicates with the external environment at an embryonic stage, and later on it either develops into mouth or develops into mouth of the organism known as prostome, and if the anus is developed, this development is considered as deuterostome. So either it may develop as mouth or anus. So it is blastopore. So option A, gonophore, is wrong. Option B, blastopore, is correct. And option D, C, oospore, is wrong. And option D, zoospore, is wrong. So option B, blastopore, is correct. Moving on to question number 35. The diagram of labia rohita is given below. Identify the parts labeled A, B, C, D, E, F, G. What is A? A we can call it as nostril. Then B is, you can see it is the eye. Then C is called as spectral, C is dorsal fin. D is caudal fin. E is anal fin. Then F is pelvic fin. Then G is pectoral fin. Is pectoral fin. So A nostril. Here also we have A nostril. Here also we have A nostril. Here also A nostril. Here also A. Go for the second one. B is eye. Here also B eye. Here also B eye. Here also B eye. Go for the next one. C dorsal fin. Here it is anal fin. So this is wrong. Here it is dorsal fin. Correct. Here also it is dorsal fin. Here also it is dorsal fin. Go for the next. Caudal fin. This is wrong. Anal fin. So this is wrong. Here it is caudal fin. Here also it is caudal fin. Go for the next one. Anal fin. Here it is pectoral fin. So wrong. So this is wrong. This is wrong. This is. So anal fin. Correct. Then pelvic fin. F. Correct. A is nostril. B is eye. C is dorsal fin. D is caudal fin. E is anal fin. F is pelvic fin. G is pectoral fin. So option D is correct. Option D is correct. Moving on to question number 36. Compare the statements A and B. Statement A. To counteract the increase in turgor pressure in plant cells, the cell wall produces an equal and opposite pressure, that is the wall pressure. Statement B. When plant cells undergo endoosmosis, they swell, but they do not burst. To counteract the increase in turgor pressure in plants, so TP, so turgor pressure. So this turgor pressure will, and this is called wall pressure. So to counteract, equal amount of turgor pressure is exerted on the wall pressure, and the wall in turn exerts the same equal pressure. So this is correct. Statement A is correct. The cell wall produces an equal and opposite pressure, that is wall pressure, when the plant cells, it is correct. When the plant cells undergo endoosmosis, they swell but do not burst. So this is also correct. So turgor pressure, as we know, it is the pressure which develops in the confined part of an osmotic system due to osmotic entry of water. It is also called hydrostatic pressure, and we know that WP, that is wall pressure, is equal to TP, that is turgor pressure, except when the cell becomes flaccid, right? So both the statements are correct, and B is the reason for A. So option A, statement A is correct and B is wrong. Then both the statements A and B are correct, and A is the reason for B is correct. Option C, both the statements A and B are correct and A is not the reason for B is wrong. Statement A is wrong and B is correct is wrong. So option B is correct. Moving on to question number 37. When red blood corpuscles containing both A and B antigens are mixed with your blood serum, they agglutinate, they clot, they clump. Hence your blood group is dash type, that is O, option A O, option B AB, and option C B, and option D A. A person with blood group O does not, as we know, O does not contain both the antigens A and B, but it contains both the antibodies, antibody A and antibody B. So when blood with both antigens A and B is mixed with a blood type which causes the blood type, then it causes what? Agglutination, because antibodies will come in contact with antigens and there is clumping. It is O. So obviously when it clots, when both A and B when with both A and B antigens, if it clots, so obviously it is O group. So as O type has both antibodies which will cause clumping with antigens A and B. So the answer is O group. So option A, O is correct. Option B, AB is wrong as it is codominance. B is wrong. Then D, A is wrong. So option A is correct. Option A, O group is correct. Moving on to question number 38. Bovine spongiform encephalopathy is a disease by prions in cow, B sheep, C man, and D potato. You know, this bovine spongiform encephalopathy is also called mad cow disease. So, And here it is given as bovine. Bovine is nothing but what? Cattle. So obviously it will be cow. So it is also called mad cow disease. So the infected cow, as you know, it causes CJD, that is Creutzfeldt-Jakob disease in human, on consumption of infected cow, when they eat the meat of infected cow. So it may lead to death of nerve cells, and even degeneration of brain takes place. So it is cow. Prions is by prions in a cow. So bovine spongiform encephalopathy is diseased by prions in a cow. So option A, cow, is correct. B, sheep is wrong. C, man is wrong. And D, potato is wrong. So option A is correct. Moving on to question number 39. Compare the statements A and B. Statement A, when the urine moves through the descending limb, it becomes hypertonic, and as it passes through the ascending limb of Henle's loop, it becomes hypotonic. The descending limb is permeable to sodium ions, while the ascending limb is impermeable to sodium ions. So in descent from the Henle's loop, when it comes to the descending loop, urine will become hypertonic because it will have more of sodium ions and less of water. So it is impermeable to sodium ions. It is impermeable to sodium ions but permeable to water. Here in ascending limb, it is hypotonic because sodium ions are less and more water, and it is permeable to sodium ions and impermeable to water. So both the statements are wrong. Here it is given permeable, so it is wrong. Here it becomes hypotonic, also is wrong. Right? So both the statements are wrong. Option A, statement A is wrong and B is wrong is wrong. Statement A is correct and B is wrong is wrong. Then both statements A and B are correct is wrong. Both the statements A and B are wrong is correct. Moving on to question number 40. To meet the demands of the society, in vitro production of a large number of plantlets in a short duration is practiced in floriculture and horticulture industry today. This is called option A, somoclonal variation. Option B, hybridoma technology. Option C, micropropagation. And option D, somatic hybridization. As we know, micropropagation is a plant tissue culture technique used for the production of plantlets. So it is micropropagation technique which is used for the production of plantlets, in which the culture of small aseptic sections of tissues and organs are taken in a vessel with a defined culture medium and under controlled environmental conditions. So this is called what? Micropropagation. So option A, somoclonal variation, is wrong. Option B, hybridoma technology, is wrong. Option C, micropropagation, is correct. And option D, somatic hybridization, is wrong. So option C is correct. Hope you have understood this part 2 of 40 questions of Karnataka CET 2010. So I'll be back with part 3. Till then, stay tuned.